Good morning, everyone. My name is Prem Mohanty. I am a life sciences product specialist and product marketing manager at Benchling. Today, I'm excited to talk about RNA therapeutics and how informatics can play a critical role in supporting and accelerating development. Benchling's mission is to accelerate life science innovation for the betterment of humanity. Our company was founded in 2012 by a team of scientists and engineers at MIT who were frustrated with the poor quality of software they had available to support their science. So with support from top-tier investors like Thrive, Benchmark, and Sequoia, Benchling built out a range of solutions for life sciences on a single platform. We are fortunate to work with over 500 innovative life science companies, including many of the world's largest enterprises. The history of biopharmaceuticals is long and exciting. Technologies such as small molecule organic chemistry and recombinant DNA technology are step changes in biopharma innovation that have led to several groundbreaking treatments. And yet, biopharma innovation is still rapidly accelerating. Scientists today have access to an unprecedented set of tools and technologies and access to all the lessons from the past. This allows them the ability to combine technologies in ways simply not possible before and to go after disease targets that have been undruggable so far. By some estimates published in literature, all the targets with drugs on the market today make up only 4% of all possible targets in the human body. So the opportunity for discovering novel therapeutics and dramatically impacting human health is massive. For the longest time, scientists believed DNA and proteins to be the only thing that mattered, and the role of RNA was just to pass information between the two. As a result, earlier therapeutic strategies targeted either DNA or protein. Majority of the drugs on the market today, whether antibodies or cell therapy or small molecules, target proteins. A smaller group of therapeutics target DNA, such as gene therapies and some small molecules. With the progress in the understanding of RNA biology, RNA has emerged as a viable third target. In particular, RNA therapeutics are being recognized as an important new category in biopharmaceuticals for its potential to prevent and treat previously undruggable diseases by targeting in ways that couldn't be addressed with small molecules, proteins, or antibodies. However, RNA itself degrades too quickly in circulation, and issues with delivery and potency limited their use initially until now. The discoveries in therapeutic categories such as siRNA and antisense oligonucleotides have been remarkable for their ingenuity and efficacy. By combining the powers of biology and chemistry, RNA therapeutics are revolutionizing how we address diseases. Chemical modifications have played a critical role in making RNA therapeutics viable across a variety of diseases. Scientists have learned to modify every part of the RNA molecule, from the sugar to the base to the linkages and conjugates. Here are some of the well-known chemical modifications that are in frequent use today. Each of these modifications confer special properties to the structure of the RNA molecule. Scientists are now able to modulate the potency, dose volume, safety, and tolerability of these molecules with chemical modifications, which helps them go after newer diseases, which was not possible until now with the native or natural RNA molecules. These have had a dramatic impact on how successful RNA therapeutics can be. Yet the potential of RNA therapeutics R&D is impeded by the current informatics tools available to scientists. Studying chemical modifications and their impact on disease biology requires an iterative and rapid discovery process. However, current tools used for oligo design are clunky and difficult to use. Additionally, there is a historic lack of a standardized language or syntax to represent chemical modifications on nucleotide sequences. Research teams spend a significant amount of time translating their bases into shared language for easier readability. Finally, oligo design data and biological assay data have been historically siloed, which meant that scientists have had to spend a lot of time connecting the two manually, often across cross-functional teams. As RNA therapeutics have gained traction scientifically and clinically, the tools to engineer them have been frustratingly limited, however. Scientists chemically modifying RNA oligos have had to contend with manual, low throughput, and disconnected software and spreadsheets to complete the full workflow of designing analyzing and testing these complex biomolecules. To date, there has not been a solution that unifies the biological and chemical nature of RNA therapeutics with seamless collaboration, analysis, and scientific insights. Data is disconnected by living across multiple systems, so contextual insights are never surfaced. Time is wasted transferring data, and the process is error-prone. This current disconnected ecosystem is not efficient or scalable. Solutions that focus primarily on chemistry workflows struggle to model hybrid chemical modifications to large biomolecules. Solutions that use a commonly available OpenHelm editor are also not intuitive for biologists. Given that there are also significant partnership opportunities for many RNA therapeutics companies, it's also important to highlight that data sets with academic, industry, and CRO partners will be siloed and extremely difficult to connect and control shared access to. Here is a little more detailed view of the specific challenges. In the first illustration to the left, the the same chemical modification is denoted by different syntax depending on the vendor you use. This lack of standardization makes it difficult to work with multiple vendors and also collaborate internally. The second illustration to the right shows the issue with ease of use while designing oligos. Most solutions are not able to handle both the sequence view and the monomer structure view of RNA molecules adeptly. The navigation between the two views in particular is often non-intuitive. Our approach to solving these challenges is really three-pronged. To solve for disconnected data, we want to centralize all your experimental data on a single platform so that you can get to the insights that matter to you more quickly. We want to standardize on a common Helm taxonomy while providing the ability to still export and interconvert nomenclature with some of the other more common vendor formats. This should help streamline collaboration both within your team and while working with external partners. And finally, we want to provide an easier and more intuitive design experience so that you can iterate on your chemical modifications faster. Let's take a closer look at each of these solutions. There are three ways in which we help you not just centralize data, but also get to your insights faster. First, we help you standardize the data that you capture across your entire team. We offer results tables, which help capture experimental output in a standardized manner. Because these results tables are connected with a data warehouse on the backend, we can centralize all results related to a sample irrespective of the user or experiment automatically without the need for scientists to look up disparate data sources. And finally, because the data is in the central warehouse, you can now query all your data from a single user interface to visualize your data. No more digging through and copy pasting from multiple spreadsheets and experiments. You can also export these aggregated data sets to run more advanced analytics and statistics outside of Benchling. Benchling provides many features specifically designed to simplify collaboration within your organization. First, we set you up with a monomer library that you can customize to meet your needs. This central library ensures that any modifications made are available to the rest of the team and also allows Benchling to run uniqueness checks and reduce errors. Second, we provide search features so that you can run either standard string-based searches using natural bases or Helm notation, or search for modified oligos based on the monomers used. Finally, for every registered RNA molecule, we automatically surface the Helm notation and IDT syntax so that you can export whichever nomenclature you might see fit. In the future, we plan to add support for more vendor notations. This means that all your work is standardized on Helm while still providing ready access to some of the most commonly used vendor formats. Finally, we make it easy to design your oligos. We provide a simple and easy to use user interface where the oligo bases are rendered in a more sequence map-like way. Modifications, if present, will appear on the sequence map using a novel visualization framework. A legend below describes the visual cues used to represent each type of modification. Each nucleotide in an oligo is broken out into a sugar, phosphate, and nitrogenous base monomer. All monomers used in an oligo, natural and modified, are represented in the nucleotides tab shown on the right side of the window. We help you standardize all your work on Helm while still providing ready access to some of the most commonly used vendor formats. Finally, we provide REST APIs to automate and increase throughput of your oligo creation and modification processes. To summarize, Benchling offers a complete solution to streamline and accelerate RNA therapeutic R&D. We make it easy to design oligos with an intuitive user interface and automation tools, allow you to better understand the impact of your modifications by connecting chemical design to biological assay context within a centralized data model. And finally, we provide the tools necessary for effectively collaborating with your teammates, both internally and externally. The Benchling Life Sciences R&D Cloud is designed to give you a 360-degree view of your R&D innovation. Benchling's applications span the R&D process from experimental design, in silico biology, sample tracking, and process management. There are 7 applications that Benchling offers, but they are all offered from a single interface. This makes it easy for users to leverage what they need without leaving the software. All of these applications are built on a common unified platform. They share a configurable data model that maps to each team's unique scientific and operational requirements. This makes it possible to query the system for sample and process intelligence using the Insights application. Benchling is built on a modern cloud infrastructure fully managed by Benchling and offers state-of-the-art security and control. And we know that most companies have a rich ecosystem of instruments, software applications, and data stores that their R&D depends on. So we provide a full set of REST APIs and out-of-the-box connectors for lab automation, as well as ways to move data onto and out of the platform. As much as these are core values across all of Benchling's capabilities, our approach towards supporting science, our customers, and innovation is particularly relevant for RNA therapeutics. We created our RNA capabilities in partnership with researchers focused on RNA to deliver on the specific needs of this hybrid modality. We will continue to stay ahead of the needs and evolve the product as the science itself continues to transform. Before we jump into the Q&A discussion, I encourage you to learn more on our website. Within the Solutions tab of the navigation menu, there is a page for RNA Therapeutics. If you'd like to speak with me directly, I welcome your emails at the email address here. With that, we can open up the forum for any discussions or questions. Thank you for joining me today and have a great day.